Hello guys, this is Shraddha Thakkar here. Welcome to my YouTube channel Biology with Shraddha. In this video, we are going to talk all about the neurons. So let's get started. Before we move on to the actual nervous system of humans, let us first understand the neurons and the nervous system present in primitive animals, starting with the Hydra first. The Hydra are animals that belong to Cnidaria. These are the first animal groups to show a true diffuse nervous system. That means they had the neuron-like structures scattered throughout their body. They also showed presence of some sensory cells along with these neurons. Synapses were also present. Now these synapses are the gaps which are present in between two neurons. The nerve cells of Hydra are scattered throughout the body, and that is what is known as the nerve net. The sensory cells are also present in the body wall and the tentacles. Let us now understand the nervous system in Planaria, that is the flatworm. It is the most primitive animal with the presence of central nervous system. The central nervous system of Planaria is located ventrally. That means it is present in the front side. It has a mass of ganglion in the head region, and there are these ventral nerve cords arising from each ganglia. The nerve cords are interconnected with each other, and we also have these sensory cells present in these worms. Next, let us move on to the neural system present in the humans. Now, the neural tissue is of two types. You can say there are two types of cells in it. First is the neurons, and second one are the neuroglial cells. Both these cells have arisen from the ectoderm, that is the outermost germ layer. Let us understand the parts of the neurons first. There are three main parts. We have the cell body, which is called as the cyton. Then there is this axon and dendron. The neuroglial cells are known as the supporting cells, and these are of four types. We have the oligodendrocytes, the microglial cells, astrocytes, and ependymal cells. So let us first understand the detailed structure of the neuron first, and then we will move on to understand the neuroglial cells. This is the structure of a neuron. So as we discussed, there is a cell body present that is known as the cyton. So here, the head region over here, this is the cell body. It contains the nucleus. Along with nucleus, there are these fragments of rough ER, that is the rough endoplasmic reticulum, called as Nessel's granules, which are present throughout the cyton. Then we have these dendrites. These are the receiving parts. So the neuron receives the impulse through these dendrites. Then this long part is the axon. Some neurons have myelin sheath present on their axon. Such neurons are called as myelinated neurons. This myelin sheath is not continuous. As you can see, there are these gaps in between, and these gaps are called as the node of Ranvier. At the end of the axon, there are these nerve endings or axon terminals which have these knob-like structures which are called as the synaptic knobs. So this was a detailed structure of the neuron. Now let us move on to the neuroglial cells. As we discussed earlier, these are the supporting cells. That means these are not involved in the direct signal transmission, but they are going to support the neurons in conducting the signal. There are four types of neuroglial cells. First is the oligodendrocytes. The oligodendrocytes function to form the myelin sheath that we just saw, and it helps in the faster transmission of the impulse. Second one are the microglial cells, which act as macrophages. So they are going to perform phagocytosis and they will clear all the dead and damaged materials. Third type is the astrocytes, which are actually star-shaped. That's why they are called as astrocytes. And these are important as blood-brain barrier. So they are going to prevent all the toxins and pathogens from entering your central nervous system. The fourth type of the neuroglial cells are the ependymal cells. Now these are the cells which actually produce the CSF, that is the cerebrospinal fluid. Let us move on to understanding what exactly is a synapse. As we discussed earlier, it is just a minute gap which is present in between two neurons, and the diameter is about 20 to 30 nanometers. It is also known as the synaptic cleft. It is the site for transmission of impulse with the help of neurotransmitters, and these neurotransmitters are especially important in case of the chemical synapse. So we have these two types of neurons. The presynaptic neurons are the first neurons which carry the impulse to the synapse, and the second one are the postsynaptic neurons. These are the neurons which receive the input at the synapse. Let us now go to the types of synapses. The first type is the electrical synapse and the second one is the chemical synapse. Electrical synapses are the ones which are really small in diameter. It is just 3 to 8 nanometers in diameter. So here the neurons are going to be very close to each other. Therefore, it leads to a faster response. And these electrical synapses will be seen in areas which are important as defense reflexes. The chemical synapse is the second type of synapse, and these are larger in diameter. They are 20 to 40 nanometers in diameter, and hence they require signal transmission to be carried out with the help of neurotransmitters, that are these chemicals which help in the transmission of signal from one neuron to another. Let us understand how exactly does this chemical synapse work. So here I've drawn this flowchart for you all for the important events involved in signal transmission with the help of a chemical synapse. So first the synapse is going to get the signal via the presynaptic neuron. So the impulse travels along the axon of the presynaptic neurons and it finally reaches the axon terminal, that is the nerve endings. The synaptic nerves that we just saw in the diagram contain the synaptic vesicles, and that's how these vesicles get activated when the impulse reaches it. The neurotransmitters which are present in the synaptic vesicles need to be released, and this will only happen when the voltage sensitive calcium ion channels are going to open up. So once the signal is received by these vesicles, the calcium channels are going to open and calcium ions diffuse inwards. Therefore, the calcium concentration increases in the cell, and that's how the neurotransmitters get released in the synapse. These neurotransmitters are then going to bind to their respective receptors present on the postsynaptic neurons. And that's how the signal got transferred from the presynaptic neuron to the postsynaptic neuron with the help of the neurotransmitters. Let us understand how exactly the impulse got transmitted into the neuron. First of all, the neurons are in the resting position, and that's when they have the resting potential as minus 70 millivolts. In this situation, the neuron has not yet received any signal. So here, the inner side of the membrane is going to have a negative charge and the outer side is going to have a positive charge. Now this is when the voltage gated channels for sodium as well as potassium are closed. Suppose that there is a signal which is coming up to the neuron. This is when action potential is generated and the potential from minus 70 changes to plus 30. This is also known as depolarization of the neural membrane. This is when the voltage gated sodium channels are going to open up and it leads to reversal of the charges. So now in the inner side we have positive charges and on the outer side there are negative charges. Next is repolarization. That means getting the membrane potential and getting the neuron back to the resting position. This is when the signal has already been transmitted. So here the plus 30 millivolts situation should change to minus 70 millivolts. And this happens when the voltage gated potassium channels get opened. So this is when we are going to get back to the resting position. That is, the inner side of the membrane will again become negatively charged and the outer side becomes positively charged. So the rate of transmission of impulse is actually higher when the neurons are longer and thicker. Thicker means that they will have presence of myelin sheath on them. So therefore we say that a higher and faster transmission is seen in the medullated nerves. Medullated means myelinated because of the saltatory conduction. Now the saltatory conduction is actually the condition in which the impulse or the action potential is actually jumping from one node of RAN wire to the another. So that was all about the neurons. I hope you enjoyed the video. Please don't forget to like and share the video with your friends and family, and also subscribe to my channel Biology with Shraddha. Thank you.